So we'll start. Hello and welcome everyone to Ferment-Home-Station. We are running this National Science Week event to promote the science of fermentation. I'm Lenny.
You're Lenny?
I'm Deepan.
I'm Lenny.
And thanks for joining us on today's DIY Lactofermentation workshop being delivered by our expert guest, Dr. Erin McKenney. Dr. McKenney is an assistant professor in applied ecology at the North Carolina State University. Her research mainly focuses on how microbial communities form over time and how they adapt to their environments. More recently, she has expanded her research to sourdough and other fermented goods. She believes that microbial cultures lie at the heart of human cultures, and fermented foods provide an accessible system for studying microbiology and nutrition without any formal laboratory equipment. And by combining citizen science and global collaborations, Erin uses research to empower students of all ages. Today, she will be combining her hobby and research expertise of fermentation and walking us through the process of fermentation, its history and the science behind it, and also at the end, showing us how to set up our own ferments. Hi, Erin, and thank you so much for joining us this Science Week.
Thanks, y'all, so much for having me. Deepan, thank you so much for finding me out in the ether and inviting me to host this webinar. I'm really, really excited. To talk to you about science, about fermentation. As you mentioned, I have been thinking about microbial ecology and microbial systems in guts and in foods for over a decade now. So I first started thinking about it in gut systems, and it's really exciting to have something that I can bring home and talk about over the dinner table, and that we can actually study without a health hazard or concern. So I'm excited for you to join me in my kitchen. So welcome. I'm going to launch right in and go ahead and share my screen. Let's see. So here's the check. Can you see it? Perfect. All right. So this is me. Today for me, today for you, we're going to be fermenting vegetables. So just a brief overview. First, I'm going to give you kind of a rundown of the history and science of fermentation. Then we're going to have, as Deepan said, a do-it-yourself lactofermentation workshop. And I'll say this now before I forget. I have some sauerkraut that I'm going to prep that'll take about 8 minutes. So that is a great time for questions. I'm also fine if you interrupt me at some point, if you really need to know something right now to clarify your understanding. But rest assured, there will be 8 minutes where I am just massaging cabbage. So there will be plenty of time for questions after this talk, before I start actively prepping and throughout. And then we'll be sharing some resources afterward as I wrap up. So to get started, again, I've been thinking about microbial systems in guts for over a decade. Really thinking about also the foods that we put into our guts and how both of those systems, whether it's the nutrients in the food that you eat or whether it's the shape of the gut that you're putting your food into, how that actually shapes the microbial ecosystems and communities that live inside you. So let's take a second, just look at your hands for a second. Look at your hands, right? You are covered in bacteria. You're covered in all different types of microbes, but particularly bacteria. There are something like 100 times more bacterial cells on you than there are human cells. To be human is to be bacterial, right? And I think a lot of us, when we think bacteria, we think germs. And I think particularly in this time of COVID and quarantine, a lot of us are starting to think of microbes as the other, the bad. So I'm really excited to talk to you about microbes in a context where we have 100 trillion superhero sidekicks living on us and in us throughout our entire lives. And I think these microbial systems are a perfect way of giving microbes a bit more of a positive name and renown. So I've been thinking about foods and guts for a long time, and they're really, really important for shaping us from our birth. Microbes shape our development. We now know that without bacteria, we would not be able to develop normally. If you live in a completely germ-free environment, you're actually in trouble. We know that the bacteria that live in our guts, as they break down the food that we can't digest, they produce compounds that actually not only nourish us, but send signals, chemical signals to our brains. So the next time you have a craving, consider it might not be your craving. Maybe it's the bacteria in your gut sending you a chemical signal that, oh, maybe that's what I want to eat right now, right? They're also intertwined in making us healthy as well as occasionally making us ill. And all of this is through that process of digesting nutritional components that we wouldn't be able to eat on our own, right? It's just incredible to me. It's incredibly humbling and awe-inspiring that these invisible multitudes are shaping so many aspects of our lives across our evolutionary history. We've developed and evolved these very specific particular partnerships with these microbes that live on us and in us. And microbes aren't only important across an individual's lifespan, they have benefited our species as humans for hundreds of generations, right? Microbial cultures are important to human cultures. People all over the world have used lactofermentation to preserve produce like fruits, vegetables, and greens for thousands of years. Some of the earliest recorded ferments, fermented produce is from 6000 BC, or before the Common Era, in the Fertile Crescent. And we now know we've been following literal breadcrumbs through time, right? A couple years ago, the earliest sourdough breadcrumb evidence is now from 14,000 years. So even before we were farming grains, we were milling it and gathering seeds from the wild and using that to prepare sourdough bread. So pretty incredibly long history, right? And this fermentation process comes in different flavors, if you will. I love a good pun. There's a lot of like multiple layers of knowledge and knowing with these systems. So when you ferment, you can think about fermentation from a microbial perspective. Are you using only bacteria? Or like if you think about a yogurt, tends to have a lot of lactic acid bacteria. We don't tend to think of yogurt as a yeasty business or a yeasty food, right? We can focus on yeast. If we're brewing beer or making wine or mead, or if we're baking bread, right? Then we tend to be very focused on yeast. Although sourdough starters have a lot of bacteria as well. I think you'll be learning about that later in National Science Week, so I won't steal the plot too much. Or we might think of these combinations of very complex communities that incorporate both bacteria and yeast. So sourdough starters, of course, or a SCOBY, right? A symbiotic culture of bacteria and yeast is what SCOBY stands for. And that would be that slimy plug that it looks like a coffee filter trying to come to life, right? That you can float on top of fresh brewed sweet tea to make kombucha. So we can think about different types of fermentation from a microbial perspective, or we can think about it from like the method that we're using to prepare the fermented food or drink. So we could think about the fermentation occurring in a liquid through brining, which is a water and salt mixture, or through a brewing perspective where we're actually heating up that liquid and we're adding a lot of sugar, or we're actually making a wort, right? Dissolving a lot of carbohydrates from grain. Or we could think about sourdough starter is a liquid of sorts, right? A very thick liquid, but it's got water in it. So it has a different kind of hydrodynamic that enables the microbes to actually interact with the substrates and with each other. Or we can think about it instead as more of a solid substrate and think about packing things. Instead of submerging them in liquid, we can think about mixing them with salt or without salt with a lot of spices, right? Or in agriculture, we actually do a lot of solid packing. It's called silage, right? So you can just pack everything down, and not even with this adding salt, you can perform similar fermentations, okay? And to the best of our knowledge, salt brines packed tightly in vessels were first used in China. And then dry salting kimchi, which is a brined mixture of produce that includes spices, originated in Korea. So this is not only a gorgeous watercolor of the world map, but also includes, just a tremendous array and variety of fermented foods that are consumed across the world. So we can see truly, no matter where you're from, no matter what your personal history is, you have roots in microbes, right? Your ancestors probably could not have survived, at least not as well, without the power of microbes. And the point here is that we've relied on fermentation to preserve and improve food for millennia. But Antony van Leeuwenhoek didn't even invent a microscope until the late 1670s, right? So we've been using microbes unwittingly for 10,000 years or more without even knowing that microbes were around us, without having any idea of what was actually doing this. And even after Leeuwenhoek discovered that there's this entire microbial universe around us and in us, Pasteur didn't research fermentation and figure out that yeasts were doing what they're doing or what they're doing, you know, to leaven our bread, to produce alcohol that helps create all of these different fermented beverages until the 1850s. We didn't start really commercializing a dry packaged yeast until the late 1800s. So really all of humanity has been unwittingly culturing microbes for, you know, tens of thousands of years, right? So why? For one thing, it's a really great way to preserve food, right? If you think about before we had refrigerators, we really were at the mercy of the environment. And particularly if you live in a warm and or a damp place, right? Warm, wet conditions are perfect for growing microbes. And many of those microbes you don't necessarily want, right? We think about if you left a jar or a bowl of food on your table or outside, you know, for a week without refrigeration, you probably wouldn't want to eat it, right? So before refrigeration particularly, fermentation was a really terrifically effective way to preserve food, to increase its shelf life and its nutritional content. So here's an example of chow chow. It's a traditional relish in the southern United States, and it's made with like the leftovers of the garden at the end of the harvest season, right? So green tomatoes that haven't ripened yet. What else do you do with a green tomato? Here you fry it, right? But you could also chop it up and put it into a jar with onions, with any peppers, and with cabbage, right? So we think about, you know, late fall, or for y'all that would be what, late spring, right? Because our seasons are flipped. So anything that's left in the garden at the end of the growing season, folks would chop up salt and put into a jar, and it would become this fermented lovely relish. So it's a way to save things that might otherwise go to waste. It's also a way to preserve them, right? So this, unfortunately, it's hard to find pictures of lacto-fermented chow chow because these days it tends to be pickled and brined instead of fermented, but still among my favorite fermented foods. And so I've said lactofermentation a few times. When I say that, I mean it's lactofermentation is an enzymatically controlled anaerobic breakdown of sugars and starches to form lactic acid and carbon dioxide, right? So to put that into pictures, I'm a visual thinker. So let's break that into some bite-sized pieces, right? So when you put this produce in a jar and submerge it in a brine mixture, it's going to be, it doesn't have a lid on it at first, right? Because you actually want it to have an opportunity to be exposed to different environmental microbes. And as they move into that jar, you've used salt, which on the one hand is kind of it's an osmotic balancing, right? If you just put microbes or anything in just plain water, then it's going to rush into the cells and might burst them, right? So you need salt for osmotic balance, but you also need salt to kind of select against a lot of different microbes that might otherwise spoil food, right? So it's a selective pressure for the more beneficial microbes that do the fermentation. Over time, this also becomes a very low oxygen environment, right? First, because it's submerged, but then because you have the growth of lactic acid bacteria, which produces acids as well as carbon dioxide. So the more microbial growth you have in that jar, as it's digesting the sugars and starches that are in your produce, it's actually producing acid and carbon dioxide that further drops the oxygen levels. So it's selectively favoring very specific bacteria to be able to grow and thrive in that jar. So lactic acid bacteria, or LAB, they are what I would consider an environmental engineer, right? So they're, as they grow more abundant over time, they're actually producing more acid, okay? So you can see, you know, again, I've graphed it. I like to think of numbers and pictures as well. So the more lactic acid bacteria you have colonizing that jar over time, because they're the ones that thrive in a salty, low oxygen environment, they're producing acid that they are able to also tolerate, right? And when they produce that acid, it drops the pH below the tolerance of many microorganisms that cause food spoilage, like mold or like the bacterium that causes botulism, right? Those things that we absolutely don't want in our food actually can't grow in a low pH or a high acidity environment. So the lactic acid bacteria are actually changing the environment to favor very specific, and in our case, very beneficial microbes. So to put this into perspective, this is like dropping from a neutral pH, right, near pure water at the beginning of a ferment to tomato or even apple juice, right? That's a pretty huge drop. And this tends to occur in just a few days, in about 3 to 4 days. Okay, so that's a pretty tremendous change in conditions in a relatively short amount of time. So this got me curious a couple years ago to wonder, do all lacto-fermented foods follow the same process, right? Does the pH drop at the same rate no matter what vegetables you use? And the great thing about fermented foods, I think I mentioned before, compared to guts, is that you can bring a bunch home to your kitchen without raising eyebrows or upsetting stomachs. Right? So I invited a bunch of friends over a couple years ago and had a fermenting party. I said, I'll provide the jars. I'll teach you how to pack this stuff. You just bring whatever vegetables or fruit that you want to pack in your jars and learn how to ferment. So I'm excited to be having version 2 of this in a global version tonight. And just like today, I told folks to bring whatever produce they want and I'd help them prepare it. But as a bonus, because they're my friends and they know me, I also sent them all home with pH paper so that they could measure how their foods changed over time. And I made a Google spreadsheet that everybody could actually edit so they could enter in their data, and the results were really interesting. So as you can see, there's a lot of variation. It looks like fermentation, the process proceeds a little bit differently depending on which types of produce you use. I didn't include the legend here because, you know, as many different colors of lines as you see are different combinations of produce. It'd be pretty messy to try and think through that on the fly. But the point here is you see a similar trend over time, right? That you tend to see a decrease in pH, but you do see a lot of variation among all, so different jars containing different combinations of fruits or vegetables tend to then grow and favor microbes that perform a little bit differently, right? Now, you will see we had 2 jars that actually increased in pH and went even a little basic. They both belong to the same friend who I followed up with as soon as I saw that. And I said, like, do not eat this, right? So, yeah, don't eat it if it goes basic. You'll notice if it goes basic, it tends to change color, like a brown color, right? You think about using lemon juice to keep your apples from going brown, right? Basic things, they will smell differently, they will look differently. One of the jars was very slimy, another one turned very brown. So I talked to that friend and said, you know, I'm also curious, like, what happened? Can we narrow down what happened with your jars? And she said, oh, I'm pretty sure I forgot to add the salt. So as we move forward, right, you need salt. It's a very important ingredient for fermenting. So I told her not to eat her ferments, but the rest of the jars turned acidic and everyone tried them and said they tasted great. So just to recap, you want to eat things that turn acidic, don't eat things that turn basic, right? That kind of disgust factor that we feel when we see really slimy foods, at least when they should be otherwise crispy or in jars, right? Or if we have a color change or specific smells, I think we've actually evolved those responses of disgust to help, keep us safe, right? So the reason that pH is so important is that it's favoring these different kinds of bacteria. Early in the fermentation process, before the pH has dropped, when things are pretty neutral, you've got any microbe that lands in that jar has access to kind of a buffet, a smorgasbord of carbohydrates, sugars, and starches that are fairly easy to digest. So you tend to see a lot of Enterococcus, a lot of Pseudomonas, and a lot of Weissella bacteria, right? So many of these bacteria live on or inside of our bodies, and a few of them can become pathogenic sometimes under specific conditions. But some of them, importantly, do produce acids. So they're some of those pioneer species that originally helped to engineer the environment. They're all generalists. They all do well in environments with a lot of nutrients that are easy to digest. And they might produce lactic acid, but they're not acid tolerant, right? So they are producing a waste product that they can't actually live with. But as the pH drops, those early microbial engineers, they engineer themselves out of living. They can't survive in the conditions that they've actually created. So this creates space for acid tolerant specialists like Leuconostoc, Lactobacillus plantarum, and Lactobacillus casei to move in and grow stable communities. And once those get in, they're there to stay. So then you tend to have these specialists that are well adapted to those very specific conditions, and they get to stay, right? So this means that fermented foods are not only prebiotics, which are foods that are full of fiber and other compounds that help fuel a healthy gut microbiome, but they're also probiotics, which are foods that contain living organisms. And since you're not cooking the things that you've fermented in a jar, when you eat them, you're actually consuming these communities of beneficial microbes. So those beneficial communities can take up to 2 weeks to develop and stabilize, but in the process, they produce not only acid, but also B vitamins and other compounds that contribute complex flavors. So we might think about, where are those microbes coming from? They're definitely coming from the environment. Some microbes may actually be in or on the produce that you're putting into that jar. And they're also coming from us, right? We are covered in microbes. And this is really important. This personal flavor, right, is so important that the most common phrase in Korean cooking literally means the taste of one's hands, right? So when you say, nobody makes it like my mom, like maybe literally, if we all harbor specific microbes on our bodies, then your ferments tonight are going to develop and have a very unique flavor signature from you. So be proud of that, own it. So with that, it's time to do it yourself. So I'm going to do a little vertigo flip here and rearrange my workstation. Stop sharing my screen. All right, are we all ready to go? You can see me. Are we good to go ahead and start? And this is the part, so I will, let's see. Yes. All right, so I've got a Pyrex baking dish that I like to put my ferments on once they're all packed, so that during the fermentation process, if they percolate a little bit, then I don't end up with drips all over my counter. That's something that I like to do. If you pack carefully, right, or if you leave enough space, you'll be fine without a drip catcher. I just tend to have big eyes and get myself in a little bit of trouble. So I'm a very enthusiastic home fermenter. You could pace yourself and stop an inch, a few centimeters from the top of your jar, right? And I'll show you an array of different sizes. So I'm working with a quart, which I think is like a liter-ish, right? I have pint jar. I have a half-gallon jar because we love sauerkraut in our house. My 2-year-old eats it hand over fist. So I'm also going to use a funnel. You don't have to. You can pack stuff by hand. And what I'm going to do, I've pre-cut, this is like the magic of baking shows, right? And cooking shows. So if you're really lucky, I'll try out my Julia Child voice later. So I've pre-chopped a big head of purple cabbage so that I can mix up some sauerkraut. I also have sliced apples. So I'll show you a fruit ferment, and I have tomatoes. So I'll show you how I pack all of these different items. So what I'm going to do is start with my cabbage. This head of cabbage was, I guess, a little bit bigger than my head, right? I didn't weigh it. I usually go by the size of the head of cabbage. For a smallish head, I would put in 1 tablespoon of salt. For this one, I'm probably going to put 1.5 tablespoons. So I'm going to start without adding any water. I'm just going to put in about 1.5 tablespoons of salt and just sprinkle it around. Now I'm just going to stand and massage this cabbage. So this is a great time for questions. So Dupont, if you want to read off some questions, I'm happy to field them.
Yeah, we have a few questions at the moment. The first one is from Roberta about how to stabilize the jar once the ferment is ready.
Stabilize, like where do I put it? What do I do with it?
I think so, yeah.
Or once it's ready to eat. So let's see, after I've packed my jars, I leave them on the countertop for between 3 days and 2 weeks, depending on what I'm fermenting. And then when it gets to a point where, like, this is delicious, I would like to eat it or save it to eat later, then I put a lid on it and I put it into the fridge. And that keeps it. Like, once it's at the point where you would want to eat it, you can kind of pause it. And because there will be acid that's been produced, that kind of helps stabilize the contents of that jar for later. And putting it at a cooler temperature really slows down the metabolism of those bacteria.
I think that answers the questions. Thank you very much. Another question from Kelsey. Do you think eating fermented products from someone else has the potential for upsetting our own microbiome, as well as potentially introducing some competition?
Oh, will you get a slap from someone else's hand flavor?
Yeah.
I wouldn't worry about it too much, unless maybe that friend who made the basic ferment, right? Like, don't eat that friend's food. But as long as it's gone acidic, then it should be favoring the growth of those beneficial microbes. So you should only reap the benefits, right? You may get some different flavor additives, but the types of microbes that thrive in a very salty environment are going to tend to be highly specialized. Yeah.
Okay, thank you.
So whether they're my flavor of acid tolerant microbes or your flavor of acid tolerant microbes, they're performing the same beneficial functions.
But just a matter of taste, if you like someone else's.
Yeah, exactly. Over time, if you swap jars, over time you might find your favorite person's sauerkraut. And to any Germans who might be in the audience, my disclaimer is I realize that purple cabbage is not a true traditional sauerkraut. I just love the color. So.
Another question from Nora. She asked regarding airlocks. Is it really necessary? On what sort of the difference between or the benefits of using an airlock and when or when not to use it?
That's a great question. I have used airlocks. I've actually set aside 1, 2, 3, 4, 5 different ways. I was thinking through, like, what can I show you tonight since I'm making all these different foods? I've set aside 5 different ways of kind of sealing your jar. Oh, here, you can, I don't know if you can see, it's glistening, right? So as I'm massaging this cabbage, the salt is drawing out the cabbage's own juices. So you don't typically have to pour any fluid over your sauerkraut. It auto brines. It brines itself. So back to the question, Laura, I have some airlock lids that I do use occasionally. Those I think are helpful if you want like a tight seal, if you're ever nervous that, you know, like if you were doing a more solid packing, then and you didn't have anything to weigh it down, you might want an airlock. Or if you don't have a tray or something to catch drips and you're a little bit overambitious, then that might help to, you know, contain even the most active ferment. I also have pickle pipes, so they're like silicone lids. I have some glass pickling stones. Before I had any of this, once people found out that I was, like friends and family found out that I was into fermenting, they started giving me all these fancy accoutrements so that I could use different tops. But before that, I've actually kept, here's your spoiler alert, I kept the outer cabbage leaves. This is what I used to use, and this is what I'll use tonight to demonstrate. And you can even use the Ziploc bag. That's actually, aside from my personal agenda to like get rid of plastic and using plastic, the Ziploc bag filled with saline is, I think, one of my new favorite ways. It's kind of a foolproof way to seal off your jar, because the weight of the solution in the bag holds the produce down and submerged. And that way, you know, if the bag springs a leak, it's just going to leak more saline. So it's not going to, you know, mess up your ferment. As you can tell, I ramble a lot, especially if my hands are busy.
That's great tips. Thank you very much.
Absolutely. All right.
Let's see, we have two more questions. Rigo asked about chopping up vegetables. So he wants to ferment cherry tomatoes and garlics, and is wondering if he has to chop up the small vegetables as well, or if he can just put them in whole.
Great question. So my cherry tomatoes, and these, I don't know, these are big cherries. I have cherry tomatoes. I'm not going to chop them up. You probably want to take the stems off, right? But I'm not going to chop them. And garlic, the only garlic I have in my house right now is already minced. But you can definitely put whole cloves of garlic in. And well done. Tomatoes with garlic and basil are one of my favorite ferments to do. The garlic you can also slice into thin slivers, so almost like slivered almonds, right? Just slivered garlic. I like to do the slivers because I'm imagining it's the maximum surface area to like penetrate your entire jar with a strong garlic flavor. But depending on how much you love garlic, you may not want that effect. But while I'm thinking about it, I also highly recommend that you save your brines. So if you make a jar full of tomatoes and it has garlic and basil in it, or just garlic in it, it's going to have the brine that's left over after you eat the tomatoes out of that jar. It's going to be full of tomato and garlic flavor. It's really incredible. I like to combine, I'll do a shot of the tomato with garlic brine and a shot of jalapeno brine. And it's like a Virgin Bloody Mary or something. It's really phenomenal. So you can also drink the brine. Let's see. All right.
Moving on to the next question. Christine asked about massaging other vegetables. So do you do only this with cabbage, or do you also, she asked about beets, or is this, when do you?
I only do this with cabbage. With beets, when I do it, I will peel the beet. I will slice it into like matchsticks or like thin slices, something that, because I just use raw beets, you could certainly do it with roasted beets if you want it to be softer. I like the crunch, but I want to cut those beets thin enough that a fork could pierce it if I eat it on a salad, right? And thin enough that I feel comfortable like chewing it. And then I'll load just those raw peeled and sliced beets into the jar with like a twist of orange peel and a cinnamon stick. Really good. But yeah, then I submerge those in a brine. So I've only ever done this massaging with cabbage for sauerkraut. And I think this is about ready. So let's see if I can show you. You can see it looks a lot darker. I've also lost a lot of volume, right? Because that salt has actually penetrated the cabbage and it's drawing out the fluids. So
two more quick questions.
Yeah.
The moment. So one is from Jocelyn about mixing whole cherry tomatoes and cut tomatoes in the same jar. That's possible?
Mm-hmm. Yes, it's possible. I haven't experimented with this before, so I don't have like personal anecdotal data to back it up, but I imagine when you cut into that tomato, you are exposing a lot of the juice and the soft insides. When I make salsa with cut tomatoes, it ferments in 3 days because the sugars are all available for bacteria to ferment. If I do whole tomatoes, I can leave them for a week or even 2 weeks. And I do get bubbles. It is an active ferment, but it doesn't turn kind of soft, right? If you ferment salsa for 3 days, it still has some body. If you do it for like 4 days, it almost turns into like a fizzy gazpacho.
Okay.
Right? So you could certainly mix chopped with whole tomatoes. You just might encounter some different flavors. It'll be interesting to find out if your whole tomatoes have less of a sour flavor because they haven't been digested as much, you know?
Okay, let's one more before we move on, and I'll save other questions for later. Just one last question for Nora for now. Very specific one. She asked about if you're adding aromatics or additional vegetables with higher sugar content, how do you know how much to add? So is there any sort of ratio, or it's just trial and error?
So this is the part that I love, like as a scientist, being able to apply this to foods at home. The world is your oyster, right? You get to experiment and do whatever you like. There's no right or wrong. I think it's more see how it goes this time and then figure out what you liked and didn't like for next time. So you should know, let's see, if you were putting just a lot of sugar, then you would really depend on the salt to keep any more opportunistic or potentially pathogenic organisms from growing, right? You would need like a higher selective pressure, right, in order to only have the beneficial microbes growing. You can certainly mix, like for a salsa, I'll use ripe tomatoes, onions, garlic. You can put in peppers, spicy or not, right? So you have a mixture of those high sugar tomatoes with the slightly lower or relatively lower sugar peppers and onions. So you can certainly do mixtures, both depending on the sugar content and just how hard or soft the vegetables are. You'll just end up with different textures. And again, you might end up with kind of a mosaic of flavors, some bits being more sour than others, just as some bits might be more crunchy than others. Yeah. Now I want to be able to follow up with all of you and find out, you know, what was your treatment in your jars?
And maybe we can send out an email in a week or two and see how people are doing. So any other questions? I know there came a few more in. I'll save them for later. And if you're ready to kind of move on, us for the process.
Yeah, so
keep listening.
Let's see. So I'm going to start with the sauerkraut because it's here. And that way I can clear my space and move on to other combinations. So hopefully somebody's doing sauerkraut as well, but also a lot of the principles will remain very similar. So I am, let's see, I've filled up the jar about halfway, and now I'm kind of making a fist and using my knuckles to really push and pack down these brined sauerkraut, the brined cabbage chunks, right? It's not technically sauerkraut yet because it hasn't fermented at all. So, oh, there was some point where I actually did this with a stand mixer, and it worked surprisingly well. I didn't do a side-by-side to see what hand flavor I might be missing out on, but I do know if you ever have your hands full, you can chop up your cabbage and whip it into a stand mixer and just set it in for 10 minutes or whatever, and you'll still achieve that auto brining. Let's see, and because this is a solid brine, this is one of the ones that I fill almost right up to the top. So I'm fearless with my fermenting, right? But also we're in my kitchen where nobody's here to see. Trying to think like how to express the amount of pressure that I guess if you were giving somebody a back or a shoulder massage and they said really lay into it, like I'm on my tiptoes behind my counter so I can really lean in to this sauerkraut. So here's a close-up of my packed jar. And I don't know if you can see from here, I've actually got liquid up to here. So it's packed really densely, but just the cabbage's own liquids have kind of filled in the cracks in the interstices. And then I'm gonna upend my bowl. So there, you see that there's a pretty steady stream of cabbage juice. And again, that's just, it was all in the cabbage the entire time, right? So I didn't add any water to that bowl. That was just a head of cabbage and a tablespoon and a half of salt. And now I'm in trouble because I'm thinking, oh, I've got time. I've got space. I can just pack and mash the rest of that into this jar. So we'll see if that works out for me. So there we go, sauerkraut in the making. I have also mixed caraway seed into the sauerkraut before. I don't know what other traditional rules I might be breaking there, but the caraway touch is pretty nice in my opinion. I really need a GoPro or something so you can see the top view here, because the brine is just right up to the top. You see it's even welling out. I can't tip it anymore because it'll spill on the counter. So that was a great cabbage, a fresh cabbage full of juices. So now we have a half gallon of sauerkraut. And you can, when you're done, you can either just set your lid just kind of askew, right? The goal is to have a lid that still allows air exchange and environmental microbes to kind of get in. But, and also, this is going to bubble, right? With that carbon dioxide production during the fermentation process, bubbles are going to accumulate in the jar. So you want those to be able to escape, but you don't want big pieces of stuff to land in, right? Or if someone sneezes in your kitchen, or if you have pets, right? You don't want their dander to float into your food. So you want something that acts as a filter, but also a block, right? And what I will do occasionally during this process is pick up the jar. And this is a little bit more difficult with a giant jar. You'll see the bubbles accumulate in the sides of the jar. And I just kind of tap it from the bottom. And sometimes I'll tip it on its side a little to let those bubbles escape. Not necessary unless you're, an eyes big kind of fermenter, like we can fit it all in there. So there's a sauerkraut. I'm going to rinse my hands so that I don't transfer, you know, that cabbage flavor. And then I know a couple of you are doing tomatoes, so I'll move to that next. Let's see. So here. I actually don't need a funnel for these. Again, you do want to make sure a lot of my tomatoes have stems still on them. So I'm just popping mine in whole, but you certainly could. I have done in the past. I've cut the tomatoes in half. Totally legitimate way to ferment tomatoes. I just like to be able to still pop them in my mouth, you know, so these will stand up a little bit more. So the jar is half full right now, and I am going to regret not having a spoon.
Big question. Did you wash the tomatoes before you used them, or?
Yes. I took them out of, I had them in the fridge in paper bags to hide them from my child because otherwise we wouldn't have tomatoes right now. Yeah, I have a bunch of tomato vines in the front yard, and I haven't had a tomato off of them because, you know, every day she's just like, tomatoes. So I hid them in my fridge, but then I pulled them out, put them in the colander and rinsed them. Yeah. So I like garlic, and the people in my household have learned to like garlic. So I'm going to do kind of a heaping, that's probably almost a tablespoon. I'm just going to put that into about the middle of the jar. Again, I haven't done this as a side-by-side comparison. I do it in the middle so that when I pour the brine in over this packed jar, I'm thinking, you know, in my head, it feels good because it's like, oh, it's going to filter everything through the jar. I haven't done a side-by-side. It's just, you know, then you can also see, instead of having like a thick plop of your garlic only in one space, right? If you're looking for a pretty jar to look at while it's fermenting, it can be a nice, aesthetically pleasing touch. Let's see. And so for these, again, since I want the tomatoes to be whole, I'm not going to jam my fist in there and pack this in, right? And the tomatoes have enough kind of heft that they'll kind of pack themselves. So I'm just going to fill this. Again, I have also done this version with, or a version of this tomato ferment with basil in it, right? So you can also add basil, and then you get that really nice herbal flavor. So I've got the tomatoes about to the shoulder, and now I need, now I'm going to mix my brine. So again, the home kitchen trick. Here I have a pitcher of water that I let sit out overnight just so that chlorine could evaporate. If you are on well water, or if you are not on, you know, like a municipal treated water that has chlorine in it, you don't have to worry about it. Also, to be honest, I have done some like urgent, we have lots of produce that needs to be used right now ferments that had water straight out of our tap, which I know has chlorine in it, and it's been okay. I imagine, so the idea behind letting the water sit so that the chlorine evaporates is that the chlorine is added to kill microbes, but here we want to grow microbes, right? So you let the water sit out overnight to remove anything that might be harmful or set your environment back, right? But again, I've done it with water straight out of the tap. I imagine that the more things you grow and ferment in your kitchen, then the more lactic acid bacteria you probably have in the air, right? Also, we know from studies of human microbiomes that female bodies tend to have more lactobacillus and lactic acid bacteria on them than male bodies. So maybe female fermenters have better luck if they use chlorinated water. But regardless, I'm going to measure out a cup of this water. I'm going to do 2 cups because I know I'm going to use it, like prepping all of this. And I'm going to add, for 1 cup of water, I'm going to add 1 teaspoon of salt. I'm using canning and pickling salt because it's not iodized. But again, you can use iodized salt in a pinch. Okay, I've done it before. Just again, the idea, I mean, we add iodine for our own benefit, but iodine is also something used to kill bacteria. So ideally, you want to use things, you know, that are not going to be detrimental to the bacteria you're trying to grow. So I'm going to add 1 teaspoon of salt per 1 cup of water, right? So here I have 2 cups of water, so I'm going to add 2 teaspoons of salt. And that mixes up to about, it's like a 0.9% saline solution. That's about the same as like your contact lens solution. That is a very low salinity compared to a lot of fermenters that I have seen, like in blogs and on the internets, right? But I'm very salt sensitive. So I've seen instructions for home fermenting and lactofermenting for up to 9% instead of 0.9%. So they're putting tablespoons of salt into their water. That starts to hurt my mouth. But I'm very salt sensitive. So if you put 3 teaspoons or 4 teaspoons of salt, you're probably fine. This is just what I do, is about 1%, and it seems to work great.
You answered 2 of the questions we had regarding salt, like how much you need to make it safe, and your experience is 0.9. Another one also from somebody.
And you can taste it, right?
Yeah.
Sorry, the other one.
The other one was also from somebody who's salt sensitive. I'm not sure how much that person used, but can you go even lower than 0.9, or do you think that gets...
I haven't tried it much lower than 0.9. I think if you go much lower than 0.9%, then you run a risk of mold being able to grow. So even with this, anything that's not submerged in the brine is going, and that's why I packed the sauerkraut in really tightly so that it would all be submerged. Anything that's not submerged, that's above water, has oxygen in the environment. So even if it's salty, mold could grow on it, right? And if it's not salty enough, you might find little mold islands floating on the top of your jar. Now, I have also found mold islands floating on my ferments a couple times. I tend to check my jars almost daily just to see how they're going because I just want to know. And I have used a spoon to just kind of skim those little islands off. And I've eaten my ferments. You should do what's comfortable for you, right? If you leave it for weeks and then you see like there's a big mold mass, then you probably don't want to eat that. But I try to be thorough and skim, you know, the entire island and to check it regularly so that they're not growing for a long time because molds can produce toxic compounds. So full disclosure, right? We make our own choices. You do what's comfortable for you. So I have this 0.9% solution. I can smell the garlic wafting out of my jar. So I'm already excited for 2 weeks from now. And what I'm going to do here is just pour it in and keep pouring and keep pouring until I have enough liquid in there to submerge all of my tomatoes. Now, you'll notice probably that if your jar is like my jar, then your tomatoes are buoyant, right? And that probably is helped along by the salt. So that's where we can use any number of implements to keep those tomatoes submerged. And I would say the same thing for the sauerkraut, right? For the sauerkraut, what I've done in the past, again, is I've kept a cabbage leaf and actually torn it to about the size of the jar. This would have been in my early grad school days, like when I was just starting to try this out. And then I might use a pickling stone, or I've done this with actual rocks, right? And I clean them first so I'm not putting dirt into my jar, but you just want something to weigh down that cabbage leaf. And that helps press everything under so it stays submerged under the level of the brine. For this tomato jar, I could either use a pickling pipe. So this is a silicone, I don't know, what does that look like? A pacifier, any number of things, right? It's bendy. So you can actually put that pickling pipe in so that you'll see, much like the nipple of a baby bottle or a water bottle, it has like a very small puncture so that gas can escape out of it, but it's still keeping enough of a seal to keep the contents of the jar submerged. You could then couple it with, or you could fill up a little bit higher so that everything is submerged. And then you could put an airlock, into the top of your airlock with, I'm trying to think of the name, pasture, swan neck apparatus, the thing that lets air escape, but it doesn't let the microbes from the environment get in. Words escape me. But you can use that more official airlock on the top of your jar, or this is where I want to show you the Ziploc trick. If you have a Ziploc bag, what I'm going to do, this is empty right now. I'm just going to put it into the jar. And now I'm going to pour a little bit more brine in it. The weight of the water holds, and I'm actually going to shut the Ziploc bag. That's mostly to keep stuff from falling in or to keep the brine from coming out. So now the weight of the brine in the Ziploc bag is pressing down the tomatoes below the surface of the brine inside the jar. So I have a decent seal, everything's submerged. I can still get gas exchange along the edges between the bag and the jar. And if I had a hole in the corner of my bag, I actually had to check a couple Ziploc bags before I selected this one specifically so that it wouldn't leak all of that saline out. But even if it did leak, it's the same saline that I added at the beginning. So that's a nifty trick if you have some Ziploc bags. Have we got other questions, or shall I move on to a fruit?
Yeah, I can ask a question. Roberta asked, how important is temperature? We are now at the moment in winter, so yeah, I think my feeling it takes a little bit longer, but yeah.
Absolutely. The same way at the end of this ferment, when it gets the way you want it, if you don't want to eat that whole jar in a day or two, in order to pause the microbial growth, you put it in the fridge, right? Because the cold temperatures slow down the metabolism and the growth rate. But similarly, in cold weather, it'll take longer. So these ferments, I will probably start to see bubbles in 2 days of carbon dioxide production, from the cabbage, maybe 1 day, because I've actually cut into the leaf. So I've exposed more of the internal juices than those whole tomatoes are exposed. So I'll probably start to see carbon dioxide production in 2 to 3 days. In the winter when I do this, sometimes it's 5 days before I see even hints of bubbles. So yeah, I just remember what season I'm in and how the temperatures are different. And then I consider it in like slow motion in colder months.
How do you determine when it's ready? People asking like, how long should you leave it fermenting?
Let's see. So my internal clock for different ferments is again, usually 3 or 4 days for a salsa. For a fruit, so the next thing I'm going to do is apples, in case anybody has fruits that they want to ferment. If it's a fruit, then I think like 3 to 4 days, because there's a lot more sugar. That's when I'll start checking it. But I will take a clean spoon and dip out a little bit of the brine and taste it. And for the salt sensitive viewers, I also find this saline, even at 0.9%, it tastes pretty salty to me, but the end product does not taste as salty. And I don't know whether that's because the salt is balanced at that point with the acid, or whether the microbes are actually utilizing the salt for something. So there's not as much salt available for me to taste. But it doesn't taste as salty at the beginning as it does in the end. Or it doesn't taste as salty at the end as it does right now. Yeah. Let's try that reversing.
One more quick question, interesting one from Beth. How much is too much fermented food to eat?
I mean, you're asking the wrong person. So let's see. There is an adjustment period if you have never eaten a fermented food, and probably if you don't eat a lot of like fiber foods or like whole produce, right? I'm thinking then there might be a mismatch between what you're putting in your body and what's already living in your body, right? So I'm gonna back up and ramble a little to give you kind of a whole picture with some background. Is that all right?
Yeah.
So your gut is your primary interface with the outside world, right? We think of our skin like I touch the outside world all the time, but your immune system isn't active on your skin. Your skin is actually a protective membrane in a way, right? It's there to, in part, to keep stuff out, but your gut, there's so much immune system components, so many immune system components that are active along the gut, because the food that you put in is a part of the outside world, right? So if you eat a lot of fresh foods that are not cooked, and or if you eat foods that are fermented, and or if you just eat foods that are higher in fiber that promote the growth of very specific beneficial bacteria, then your immune system is always talking to a very diverse crowd, right? But if you are not eating a lot of diverse foods, if most of the foods that you eat are generally cooked or highly processed, then your immune system will be trained, it'll be seeing a different world than the world that we live in, right? Does that make sense? It'll be seeing kind of a muted, a deadened version of the world, because when we cook things, we kill them, right? We're sterilizing it. So if you go from a more muted or sterile view of the world to all of a sudden, I'm eating live probiotics, whoa, your immune system might not quite know what to do with it. It won't see beneficial microbes because the way that your brain knows, it'll just see there are a lot of bacteria. Maybe I should get them out. So some people who go from not eating much live food at all to eating a lot of live food all at once, it's kind of a shock to the system, and your body might try to flush that out. And that's a euphemism that hopefully y'all are getting, so that I'm not talking about bodily functions on a home fermentation workshop, right? So there's a shock to the system component. You might want to ease into it in the same way that people might recommend that you not eat 17 prunes a day, right? But perhaps unlike prunes, you can condition your body to then recognize, oh, this is living food, and I actually like the critters in this food, right? It'll learn how to get along in the same way that, like, post-pandemic quarantine, we might have to ease ourselves into like the regular pre-COVID social life that we had before, right? It might feel like a bit of a shock, like there's so many people. Yeah, so I think it's an easing your immune system into recognizing that these are wonderful foods and these are wonderful microbes in those foods. Beyond that, if your body is used to eating a diverse, live or a raw or a fiber-rich diet, the only instance that I've heard that there's just too much fermented foods is for people who are taking helminths that are self-treating with like a tapeworm or a hookworm for therapeutic reasons, which is an entire other scientific story. Those helminths don't get along very well with a huge influx of lactobacillus bacteria or lactic acid bacteria. That's the only instance that I've heard where if you were dependent on taking therapeutic helminths to calm your immune system down, then you might not want to eat a whole jar of lacto-fermented foods in one sitting. Yeah, otherwise, go for it.
Long answer. Thank you very much.
Yeah, exactly. Sorry, there's always that danger with me. So the last thing I'm going to do is take these pre-sliced apples and just put them in a jar. It's a lot of the variations on a theme, right? And in this jar, I'm actually going to add some cinnamon sticks because cinnamon apples, right? I'm going to put 2. Now, I used to use ground cinnamon for some of my beet fermentations when I first started. I do not like ground cinnamon because it makes a very thick slurry, and I think bacteria are able to colonize those small particles of ground spices, and it sometimes was thick, almost slimy. So I just use whole cinnamon sticks. They were otherwise perfectly delightful beets. They just were a little slimy at the end of the jar. I'm going to put, I'm excited about cinnamon, so I'm going to put 3 cinnamon sticks in this jar. And again, for presentation's sake, if you want to like look at your table and be like, wow, I am growing this and it looks gorgeous, right? If you want to Instagram it or tweet it out, you can like set your sticks up to like be against the glass, or you can just set them in the center. I mean, whatever. They're in there. That's what counts. You can also put a twist of orange peel if you want, kind of an orange flavor. You could also put some clove or nutmeg in there, right? I just don't have those things readily on hand. So I'm gonna, it's the same thing, pack it. And then.
All these apples, or is there a reason for peeling apples when you ferment them?
The apple, like the flesh of the apple softens a bit as it's being digested in the jar, and the peel is not softened as much. And these, they're actually, it can become like a tart apple mash. So if you're going for an applesauce or something, just the peel holds up. So the peel might end up feeling, I feel like if you're eating a crunchy apple with peel on it, then you don't mind the peel because you're biting through everything anyway. But for me, there's a difference in texture that I am not a fan of. But it would be kind of like maybe eating the rind of cheese, right? If the rind of Gouda were really thin. It's fine. It's not going to hurt. I'm just exposing all of my apple to fermentation, and I'm going for a uniform texture. And now this time, instead of making a saline solution, and there's a disclaimer here too, because I'm full of disclaimers. Instead of a saline solution, I am going to add lemon juice. Now, the lemon juice is going to immediately drop the pH, right? Whereas with these salt brined concoctions, I'm waiting for the lactic acid bacteria to produce the acid that will drop the pH over time. So that's maybe a slower process. Because the apple, and I've done this also with blackberries. I have a friend who did it with blueberries. So you can do it with other fruits. Because those fruits are much higher in sugar and in simple starches, easily digestible foods compared to fiber, right? I'm trying to kind of push a more selective environment from the very start. Does that make sense? So I'm going to put like a tablespoon and a half of lemon juice. Okay, so that's going to be a pretty low pH. Now, my disclaimer here is I have seen blogs and instructions online that include a little bit of lemon juice and salt in apple ferments. So it's perfectly fine to put salt in apples. just not what I have done, but my way is not the only way or the only right way to do it. So, and again, it's our same step. We've prepared our, well, not a brine in this case, but maybe a light acid bath, right? Until I have just enough that those apples can stay submerged. And then I'm going to use a pickle pipe. So I'm going to bend it down. But again, you could use, I've done this before with like the top of an apple. So I've kept the peel on like, I've cut like the top, I don't know, a couple centimeters, right, a cross section of the apple so that it fits in the same way. And it kind of weighs down the rest of the apple below the brine. So that's the goal. And again, over time, you can see some bubbles. I don't know if you can see those bubbles. There are some bubbles in that jar. So, you know, your most thorough way is to, again, tap it. You're burping your jar. So you can do this right now if you have a ton of bubbles. You may want to do this depending on how active the ferment is as it goes. You may want to pick it up and kind of burp your jar periodically. Just particularly if, like me, you like to fill your jars all the way up to the top so that they don't percolate and, you know, ooze across your counter or fill your Pyrex catching pan. So that's what I've got. Bring on more questions.
Yeah, I think we are trying to start wrapping this up. So if anyone has any more questions, I have two more. But yeah, if people have questions, please post them now, and then we try to finish the session. So the first question I have is, any fruits or vegetables you don't recommend for fermentation in terms of health, texture, taste reasons?
Let's see. So the ones that I have done and that I've been quite happy with have been apples, again, blackberries, tomatoes, the cabbage. I've done the tomatoes that are red or green. I've also done radishes. So you can do whole, like small round radishes, or I've done daikon radishes, like one might use for kimchi. I've just done daikon radishes in a jar. The friend whose ferments went basic used jicama in one of them. I think perhaps part of the trouble was definitely that she forgot to put in salt. Another thing might be that, you know, jicama is a root, but it's very sweet. So I think there just might be a lot of sugar there, but maybe with salt, it would have been fine. Okra, I haven't managed to do that well yet. Like well enough that I would recommend it to someone else. I love pickled okra, like that you buy in a store made properly. I've made pickled okra, like lacto-fermented it, and it just got really slimy, not because it went basic, but because there's a lot of pectin in okra. So the same way that it can get slimy if you just saute a bunch of it or put it in a soup, it just was really slimy in the jar. So for a texture for me, that was a little off-putting. Yeah.
Great.
Oh, and cucumbers. You can put cucumbers into a jar and do this too, instead of, you know, making canned cucumbers. I highly recommend that. Yeah.
Okay, thank you very much. Any recommendation on what kind of apples you use or which you prefer using?
I am partial to like a very firm apple, just in life. I like to eat baking apples as like regular snacking apples. I am not a fan of the red delicious, the yellow delicious, or golden delicious. I think those are misnomers. I don't think that they're delicious anymore. And Fuji and Gala, I tend to find to be very sweet and sometimes soft or mealy. So I don't like the softer apples, but that's also my personal preference in life. But I find that the fermentation process does soften whatever you have put into that jar. So if you start with something firmer, it's less likely to fall apart.
Okay, two more questions. I think one we already touched on earlier. Just to clarify, there's a question: when do you put the stuff you put in the jars? When do you put them in the fridge?
Yeah, it depends, like when you want to eat it, right? So I've left sauerkraut on the counter for 2 and sometimes 3 weeks before. Just I lost track of time, and like that goes to show you how shelf stable it is at that point, right? That it's okay. The tomatoes will probably be ready to go in like 7 days, maybe in 5, just depending on how active things are here. For y'all, again, if you, since you're in winter, it might be a little bit longer, right? What might take me 5 or 7 days might take you 7 to 10 days. But again, you know, you can definitely taste this. You can either put your finger in and, you know, a clean finger and just bring up some brine, or you can put a clean spoon in and dip out a little brine and just see when it tastes good to you.
Great. And the perfect last question. People want to know if you have a blog or personal site for more information.
Oh, I do. My site is called Do Teach Learn, and I can, let's see, shall I pull the computer up and type it in?
Otherwise, we will send out an email after the session with sort of links also to your site, so people will provide this link
Perfect.
to them. Yeah. Yeah, thanks everyone for your great questions. Erin, do you have any last words?
Thanks so much. Yeah, I went way over. This was a ton of fun. Thanks for your questions. Thanks for joining me. It feels really good to be connected, and I love being connected with Australia again. So it's been a great treat.
Thank you. Again, thanks to all the participants for joining us. Thanks, Erin, for sharing your wealth of knowledge this Science Week. And especially at a time where we are all fermenting a lot now that all over the world, it's great to connect on that common theme. We would love to share any pictures and information that you got that you're going to get from your setting up your ferments. So if you've got anything that you want to share, please share it on Facebook and tag us or send us an email. And we will definitely be sending an email out with maybe some of the recipes and a site and a link to Erin's site where you can find more information.
Perfect. Thanks so much. Happy Science Week.
Thanks, everyone. Yeah.
Good night, Erin.
Bye.
Bye.